Good morning. I'm Amy Lensing-Tate, the Executive Director of the UWM Alumni Association. Welcome to our Mobile Master Chat. We're thrilled to have over 100 alumni from across the country with us today to hear from Dr. Allison Donnelly, Associate Professor in the Department of Geography. Allison completed her master's and PhD in environmental science at Trinity College in Dublin, Ireland, where she focused on the impact of air pollution on pine trees and examined the response of wheat to elevated concentrations of carbon dioxide and ozone. She spent a number of years at the University of Nottingham in the UK as a postdoc, where she continued working on the impact of climate change on agricultural crops. This is when she became interested in phenology and how it could be used to detect the impact of increasing temperature on plants and animals. At UWM, she leads a team of students whose research interests include plant and bird migration phenology and satellite remote sensing. She's collaborated with colleagues from around the world and published extensively, including in journals such as Climate Research, Global Change Biology, Agricultural and Forest Meteorology, and more recently in the International Journal of Remote Sensing. I'll join you again at the end of the chat for audience questions, but now I'm happy to turn it over to Dr. Donnelly.
Thank you very much for that lovely introduction, Amy. And I'd like to also thank the entire team, to Kyle and to Abby, for organizing this event and for inviting me to talk to you today about monitoring Downer Woods. So before we launch off into space, I will be talking about monitoring Downer Woods from down here on the Earth. And before I get to that, I'd like to introduce you to our team. First of all, we have Chloe Rayberg. And Chloe was a master's student on the project, and she was monitoring the shrubs of Downer Woods. Dr. Rong Yu is the postdoc on the project, and Rong is responsible for the extraction, the analysis, and the interpretation of all the satellite data. So before I came to the US, as Abby was just saying, I was involved in and very interested in how vegetation responds to a changing climate. So when I came here to the geography department, Rong was working on the impact of climate change on the growth and development, or the phenology of trees in Downer Woods. And she was working with Professor Mark Schwartz on her PhD at the time. So I was very, very keen to start working with Rong. So what I want to do today is to first of all, find out how to change the slide. Okay, sorry about that. So what I want to do today is to talk a little bit about phenology, determine whether it's important or not, give you some examples of how we define phenology and how we determine it, and then relate that to what we're doing in Downer Woods and the different monitoring we're doing in Downer Woods. I want to talk a little bit about the shrubs. When you think of a temperate deciduous forest, you tend to think of the trees, but we decided we were going to go for the shrubs because they're kind of understudied. But if you think of it, at the very extremes of the growing season, in early spring and late in the autumn, the shrubs are photosynthesizing while the trees are still kind of leafless. And then I want to talk a little bit about the differences between the native and the non-native shrubs. So we have native and invasive species, and they have different phenological profiles. And then I want to relate that to our Venus project, which is the satellite data. So we're trying to see if the satellite data can see the greenness. It doesn't really take photographs of the vegetation, but it gives us an indication of greenness. And if we're talking about greenness on plants, we need to talk a little bit about chlorophyll. So I just mentioned that the shrubs stay greener longer in the season, but are the shrubs photosynthesizing? And then I'll talk a little bit about why we see differences in what we're monitoring on the ground when we go out into the woods and what we're seeing from space. So phenology. Every single plant and animal and organism on the planet develops and grows. Dormant bud is going to develop into a leaf. A caterpillar is going to develop into a moth or a butterfly, and a chick is going to hatch out from an egg. So we're very familiar with these different developmental stages. Well, phenology is simply the study of the timing of those phases when they occur. So is it important when a bud starts to develop into a leaf and photosynthesize? With climate change, we have carbon dioxide in the atmosphere, and once we have these the leaves coming out and photosynthesizing, they're taking carbon dioxide out of the atmosphere. So different ecosystems will be leafing out at different times and taking carbon dioxide out of the atmosphere. So if we know when this is going to happen in spring and when it's going to finish in autumn, we can measure the amount of carbon being taken out of the atmosphere. So we can do a carbon budget. So that's kind of important if you're talking about climate change. And with climate is changing, we'll see these events occurring later in the season. Okay, so I just mentioned that phenology is the study of the timing of these events. We can use phenology to determine the timing of the different seasons, the timing of spring, the timing of autumn. So we're talking about the timing of recurring life cycle events in plants and animals. And these life cycle events or growth stages or developmental stages are also called phenological phases or phenophases for short. Some examples include things like bud burst. So we have a dormant bud here, it develops through bud burst, and we have the leaf emerging and leaf unfolding and leaf fully unfolded. So we have this progression throughout the spring season. We have things like the timing of flowering, the timing of fruiting, and in autumn, we have the timing of leaf color and leaf fall. So we've lots of different types of phenological phases throughout the growing season. Animals also have phenological phases, and just a couple for examples are like bird migration dates, the time of arrival of birds, the timing of egg laying and hatching dates in birds, and then of course the timing of departure in autumn as well. Appearance times of insects. Insects will appear early in spring, fly during the summer, and then they head off in the autumn. Other environmental or other phenological phases include things like ice melt dates. Now it's not biological, but it occurs at the same time every year. So the ice melts in spring and then forms again in autumn. So what do all of these phenological phases have in common, and this growth and development throughout the years that occurs at roughly the same time? They're driven by something in the environment. So something triggers a dormant bud to develop into a leaf. Something triggers birds to migrate, insects to appear, and ice to melt. And that is, surprise, surprise, temperature. So as the temperature warms in spring, all of these events will start to occur. Now, if the temperature gets warmer and warmer every year because of climate change, we would expect these events to start to occur earlier and earlier in the year. So the timing of these events is critical and is influenced by changes in the climate. Okay, so monitoring phenology and the use of phenology in our everyday lives that we don't even think about. Phenology allows us to predict and address any hazards in the environment. Now, it's not going to prevent them from happening, but we know when it's going to happen, and then we can prepare and put some mechanism in place to reduce any of the risk. So examples of this can be wildfires. If a forested area is very dry and there's leaf fall and leaf unfolding, we can see that that can be the time when a particular ecosystem is vulnerable to fire. Pests and diseases. The phenology not only of the crop, but also of the pest needs to be synchronized for damage to occur. So we will know the timing of leaf unfolding, like when leaves are unfolding in temperate deciduous trees, they're more susceptible to damage from late frost, or from insect damage, or from other diseases. So we need to know the timing when this occurs, and then again, we can put mechanisms in place to reduce the impact. Phenology also helps us to predict and to manage invasive species. So in this example here, we have a very dry environment. The plants respond to rainfall. So if there's a rainfall event, what you might find is that invasive species, they will respond much faster than the native vegetation. So if that happens, it gives us an opportunity to manage those species, because, for example, if they start to leaf out earlier, we might be able to use a herbicide to try and control them. So it helps us to predict and to manage natural hazards. We plan many of our cultural and recreational activities around phenological events, such as this beautiful cherry blossom blooming in China or Japan, or even in Washington, DC. We also plan our ecotourism around many events, such as this one here, we have whale watching, but also we might go bird watching, we might go caribou watching, or even wildebeest watching. So a lot of naturally occurring phenological events will help us plan our recreational activities. And of course, we're very familiar with wildflower displays in the desert, or with color displays in northern Wisconsin or Maine, or wherever you go to see your fall colors. Something that's very topical at the moment is the flu season. The viruses also have a phenology, and we're susceptible to viruses at different times of the year, and the flu season is very important to us. So this is a human health issue with phenology. So we know when the flu season is going to occur. It occurs at roughly the same time every year, and we need to plan for that. So the hospitals need to prepare, doctors need to prepare, we need to prepare. So it helps us to plan our activities. The allergy season, everybody that suffers from hay fever is well aware that it occurs at the same time every year. And what we're seeing now with the pollen season is it's starting earlier because of climate change, it's lasting longer, and it's more intense. So again, phenology is not going to prevent these things from happening, but when we know, we can put plans in place. We cannot talk about agriculture without talking about phenology. Every single activity in agriculture is determined by the phenology of whatever crop you're trying to grow. And just a quick example, because I was kind of interested in wheat, there's a growth stage called growth stage or phenological stage 32. And that's when the ear of the wheat forms in the plant when it's very, very young. And that's a critical stage because that will determine ultimately the yield of the wheat. So once growth stage 32 occurs, the farmers need to apply fertilizer at that time because we need to optimize the yield in future. Now, if you apply the fertilizer before growth stage 32, the plant is not ready to take up that fertilizer and to use it. If you apply it after growth stage 32, well, then it is no use to the plant because the ear has already formed and been established, so there's nothing you can do. So the timing of lots of agricultural activities is determined by the phenology of the particular crop that we're using. Okay, so lots of uses of phenology. And one of the main uses of phenology, and phenology has become so important in the last couple of decades, is because it's easy to understand. We can easily demonstrate to policymakers the effect of climate change at the local level. Like climate change is like on a global scale, but we can see it, we can observe it in our back gardens. Now, mind you, we'd have to have a long time scale, but nevertheless, we can do that. And phenology, or the impact of climate change on phenology, is very easily understood, and it clearly demonstrates the link between warming and the natural environment. And it was the Intergovernmental Panel on Climate Change fourth assessment report that really started to use phenology to demonstrate these impacts. And we used a summary of more than 100,000 different phenophases from 21 European countries. Of course, Ireland was involved. And we showed that 78% of the timing of leafing and the timing of flowering were earlier over a 30-year period. So we had evidence that rising temperature was having a detectable impact on our plants and animals growing in the wild. So we had shown that this earlier timing was driven by increases in spring temperature. So I can hear you all in the background there saying, oh, right, okay, so what about the other 22% of species that didn't respond? And this is what people can latch onto and say, well, not everything is responding to climate change. Exactly. We would not expect every single plant and animal on the planet to respond to climate change in exactly the same rate. And an example would be if you took all of us, we're all the same species, hopefully, and put us out into the bright sunshine for a couple of hours. Same environmental stimulus, we're all adults, we're all the same species, bring us back into the lab and look at the impact of that on our skin. Some of us would get freckles, others would go beautifully brown, somebody else might go bright red, might have no effect on other people. In fact, we would be more surprised if the two of us that get freckles get exactly the same number of freckles in exactly the same place. So that's only one species, you know, all the same age. We would be shocked if two people had the same response to the same environmental stimulus. So that's why we don't expect, in fact, if anybody shows you a study where everything is behaving in the same way to climate change, I would be questioning it a little bit. Because everything in the environment is natural, when a tree starts to bud burst, it doesn't go bing, all in one day, all the buds come out. It's an adaptation strategy that you'll have a few buds here, a few buds there, because if there's a late frost, it won't wipe everything out. It might set it back a little bit, but we have this natural variability built into the system. Okay, how do we determine phenology? You know, it's a simple thing. Well, it is simple to understand and to record, but it has to be, the data that you're collecting has to be accurate. Now, we can look at phenology in a number of different ways. One of the ways is in situ observations. In situ, you go out into the field and you monitor what's going on, which is what we're doing in Downer Woods. We go out, we observe at what stage the different plants are at. In Europe, we have a number of different professional networks where it's part of somebody's job to go out, and it's usually located with a Met office. So we would have a Met station, a meteorological station or a weather station, and we would grow certain trees at that location. So we would have temperature data and phenology data in the same location. And we get things like this. Now, I know this is a tiny little graph, but I just wanted to show you that over time, this is the timing of when leaf unfolding occurs at Valentia Observatory, which is down in the southwest of Ireland, for beech trees. And this is just showing the day of the year when it occurs. And we can see that over time, this 40-year period, 40 or 50-year period, we can see that leaf unfolding is occurring earlier in the season. And that is driven by spring temperature, rising spring temperature. But with phenological data, you know, it's kind of tricky because we have gaps in data. We have this huge variability. And that's why we need a long time period. The longer time period we have, the better, and the more accurate our interpretations and our trends will be. Because if we looked here from like 1996 for the following 10 years, we looked at that data, it looks like the leaf unfolding is occurring later in the season, which is contrary to what we would expect, but as dictated by climate change. So that's why we need the longer term trend. And just incidentally, leaves are bud bursting about 3 weeks earlier now than they were in the 1960s. This is just a picture from the area. Another way to collect the information where we can get a much broader perspective of the environment is through citizen science. When you have one scientist or one professional observing just a few trees, you know, the spatial coverage is not great, but we have very accurate data. So we try and get citizen scientists. I mean, you've heard of plenty of citizen science projects to try and collect information in the environment that can be robust enough to be used scientifically. So we get scientists to go out and observe what's happening in the environment, and they can record those observations in the National Phenology Network website. So we have the time series for this is not as long as it would be in Europe. There are very few long-term data sets in the US, but we have a very, very broad coverage. So we have different things in different locations. Another way to collect information is through, or phenological information is through remote sensing. Remote sensing just means at a distance. We can use satellites, we can use phenocams. A phenocam is just like your webcam, except we have a phenocam. It's just observing the phenology of Downer Woods sitting on top of one of the Sandberg buildings. And this is the kind of information we get. So we can get a whole landscape from satellite data, PHENOCAM is a bit smaller. So this is just the information we get from a satellite for Downer Woods. This is EVI, it's an enhanced vegetation index. A vegetation index simply gives you information about the vegetation. So, and it's divided into these tiny little boxes or these little pixels, so we can get very fine detail about the greenness. So the cameras are, the satellite sensors are measuring greenness, if you like. I'll talk a bit about this in a minute. And here we have a picture from the PHENOCAM. The PHENOCAM takes an image every 30 minutes. And again, it measures the greenness as well. And we can compare the resolution of these two images. And we can track that over time. We can see how it greens up in spring, stays green during the summer, and then browns down in autumn. So we've in situ observations. We need individual people to go out and observe it. So it's very labor intensive. We've remote sensing, you can kick back and get the satellites and the PHENOCAMs to do all your work for you. And then we also have modeling. We can take a particular species and we can model the timing of bud burst of birch, is the one that we have here, for example. And we know that temperature is driving bud burst, so we can drive the models with future predictions of temperature and see how that will impact the timing of bud burst in future. So, for example, we're just showing here that when we get up to 2060, the timing of bud burst of birch is going to get earlier. Now, it's not getting earlier at the same rate at all locations, just because there's huge variation within the different individual plants. Okay, the moment you've all been waiting for, a brief history of Downer Woods and our involvement in Downer Woods. First of all, here we have Downer Woods in 1964 and again in 1971. And this white box is just roughly the size of Downer Woods today. I didn't put an accurate measurement here, but that's roughly the size of Downer Woods today. So you can see that once upon a time, Downer Woods was part of a much more expanded and extensive woodland. And in fact, around about 1921, it was still part of a farm. Downer Woods itself was gifted to the Milwaukee Downer College by the Pfister family, and the area of the woods was around about 30 to 35 acres at the time. And today it's only 11.1 acres or 4.5 hectares. It was transferred then to UWM in 1964, and the Board of Regents and the Planning Authority had approved the removal not only of the woodlands, but of all the Downer colleges for the rapidly expanding new UWM complex. And you can see here the Sandberg complex here really encroached on the woodland itself. So they were about to get out their saws and take down the woodland until Dennis Conte came on board. And Dennis was part of the state legislature at the time, and he was the chair of the finance committee. Now, Dennis is a huge defender of the environment, and at the time he was appalled at the decision to remove Downer Woods completely from the face of the earth. And I have to say, we're very glad that he jumped in and he overturned the decision and put a preservation order in place on Downer Woods way back then, so like nearly 50 years ago. And here we are today with this amazing resource. Not only do we use it as a teaching resource and research resource, but this is now part of a globally important international satellite observation network. So Dennis, I think Dennis is with us on this Zoom call. Thanks for providing this resource for us, and hopefully we'll have a chat with you later on. I had the honor of talking to Dennis before Christmas. Actually, he saw that this talk was coming on and he contacted me and we had a great old chat. So thanks again, Dennis. Okay, so here we are in Downer Woods. Why do we look at the shrubs? I can't talk about everything today, but we just decided to look at the shrubs because temperate deciduous woodland, everybody looks at the trees, but as I mentioned earlier, we feel that the shrubs are important at the very start and the very end of the growing seasons. They're kind of the overlooked forest layer. They're not well studied. They're a bit more tricky than the trees. I'll show you in a moment why. But you can see here that leaf out, I mean, they're fairly green at this stage, but the trees are leafless, and even the ground vegetation. The ground is still pretty cold at the very start of spring. So we wanted to look at the shrubs because, well, because of this earlier timing, but also we wanted to look at the difference between native and non-native species. So what we observe, you know, not surprisingly, but we need to quantify these kind of things, that the shrubs leaf out in spring before the trees because they have to get going before the canopy closes over. And this has implications for the birds and insects that depend on the shrubs for their habitat and also for their food. And we also wanted to investigate the difference between invasive species and native species. Now, you don't tend to get native or invasive trees because the shrubs are lower growth form. They tend to there's a better chance of them becoming invasive than the trees. So a lot of things going on there with the shrubs. In late autumn, the shrubs stay greener for a longer time period. Now I'll talk a little bit about, they might be greener, but are they photosynthesizing and taking carbon dioxide out of the atmosphere? We'll have a look at that later. So here we have a group of students in the biogeography class standing in front of this huge area. This is completely green, this buckthorn. So this is an invasive species in Downer Woods, and it's very, very green. All of the trees have lost their leaves. The shrubs, the native shrubs have already lost their leaves also. There are a few oak in the background, they're marcescent, they hold on to their leaves even though they're colored. And the idea behind the phenology of invasive species is it gives an opportunity to manage the invasive species. So you could go out and use a herbicide to kill off the invasive species while everything else is dormant. Now, it doesn't always work, but anyway, that's the theory behind it. Oh. Next slide. Right, so this is just an example of what we're doing. This is Downer Woods here where we're located. And this is an image of the woods. These dots here are just the location where the trees are actually monitored. We can't study the shrubs without the trees just because they're interlinked, and we want to get an image of the entire ecosystem. We also monitor things like light, what light levels are like, air temperature, relative humidity, soil temperature, and different environmental factors. Now, the phenocam is up here on Sandberg, and it looks down on the woods. Again, we can get information from that. So what we have here, what we monitor is the timing of when bud burst occurs, the timing of when leaf out occurs, the timing of leaf coloring, and also of leaf fall. And the shrubs that we monitor them every other day. We have 5 native species. I only have 4 of them here. Wild currant, cranberry, nannyberry, dogwood, and the other one is chokecherry. And then we have 3 different invasive species. We have the honeysuckle, the buckthorn, and the European privet. So every other day we go out and we determine what phenophase these species are in. So we go out spring and in autumn. Now it's intensive, it's labor intensive, but we need that data. And the reason we go out every other day is because that's when the Venus satellite goes by every other day. And we're trying to match what we're seeing on the ground with what we're seeing from space. We also have trees, the usual suspects, a couple of oak, basswood, and ash. So this is kind of the community of shrubs and trees that we are monitoring in Downer Woods. So what could possibly go wrong with monitoring shrubs every day or every other day? So when I went out to Downer Woods this autumn just gone by, after going, I could see that a lot of our species, we lost 4 species, which is not great if we have only 5 native species. But I'll tell you about that in a second. So first of all, we had a dead tree falling on our wild currant patch. You can see some of our labels underneath the dead tree. We had mold occurring on the nannyberry. We had massive insect damage on the cranberry bushes. And then we had the chokecherry suffering from an early frost. It just got zapped immediately. Now, these are natural events. You know, this is what happens when you're doing field work. It's just a bit unusual, get 4 of them wiped out in the 1 year, but it's not the end of the world. If it's trees, usually we try to monitor the tree is going to be there for 50 or 100 years, so we need to monitor the same tree over and over. But we just selected representative shrubs at roughly the same age and same location in the woods and kept going, because we're just looking for the overall phenology. It's not hugely important that we monitor the same individual shrubs. We try to do when we can, but we can't lose all our species and just forget about the study. So that's one of the hazards of doing field work in the environment. Another thing, we have the ash trees. Again, this was one of the trees that we were monitoring. And you can see very, very few leaves left on this tree, whereas all the others are still very green and still loads of foliage. So again, with this, we still monitor it, but we can't use that data in our analysis because it's not really representative of the woodland. And probably emerald ash borer is responsible for this. So take a deep breath. I'm going to go through this slide. It is all of the phenological data for the shrubs that we have so far. We only have 3 years data. We have non-natives or invasives, and we have native shrubs. And up here, we just have the community of shrubs. The green represents spring. So this is the start of spring, the start of bud burst, and the end of spring, the end of leaves fully unfolded, like completely out. And then we have the start of autumn and the end of autumn, sorry, the start of leaf color and the end of leaf fall. So we have the spring season and the autumn season. We have 3 years data so far. Not enough, well, we have, yeah, it's not great. It would be better if we had a much longer time series, but look, this is all we have for now. We can still see variation. In 2018, you can see that bud burst started much later than the others. We had a cold spring, so bud burst started later. But we've seen that once bud burst starts, it goes very quickly. And we've seen that also in trees. A late spring means green up will occur at a faster pace. But then if we look down at all these are just the individual species. You see variation, you know, some very, very short bud burst periods and some very long leaf unfolding periods. So we see variation. But in all of these spring between the non-native and the native, you don't see a huge difference in the two groups. Now, there is a tendency for the non-natives to leaf out a little bit later in the season. So the natives are starting earlier. Now, it's only a tendency. It's not statistically significant yet, but I reckon if we had a lot more years, we might see that divergence. It's a good strategy to leaf out a little bit later because then you're not going to be zapped by a frost. So we're seeing that the invasives might be leafing out a little bit later, but look at what happens in the autumn. Very, very clear divergence between the non-natives and the native shrubs. The non-natives, the invasive shrubs, are staying greener a lot later in the autumn than the native shrubs. So they start to yellow or they start to color later, and they keep that greening for a lot longer than the native species. So overall, we can see that the non-natives or the invasives, they leaf out later, they reach full leaf faster. So once they leaf out, they go a lot quicker than the natives. They catch up really, really quickly. And the leaf fall occurs much later in the season. For the natives, the leaf fall starts earlier and they have a shorter autumn season. I mean, that's clear, you know, just from the length of the bars here that we can see. Now, I didn't show you the tree data on this as well, but just to let you know, the trees tend to leaf out a couple of weeks later than the shrubs, and they lose their leaves a little bit earlier than the shrubs as well. So that's kind of the data. That's our in situ data. So moving on then to what we're seeing from the data. So how we got involved in this project is a bit mad, really, because in about 2014, there was this call sent out to scientists all over the place. Did you want to get involved with this new microsatellite? And they were looking for people who could help them validate some of their sensors that were going to be on the satellite. So Rong and I decided, well, should we have intensive data for Downer Woods? Let's see if we could validate their phenology products or their phenology sensors. And so we heard nothing. So we kind of thought, oh, well, there you go. At least we tried. 3 years later, we got an email to say that Downer Woods had been selected as one of these important sites. And out of hundreds of other applications that they have, they selected ours as one of about 120. So we were absolutely thrilled. And this is actually the launch of the Venus satellite. And actually my brother's friend was at the launch of the satellite. But that's a story for another day. Anyway, it's called the Vegetation and Environment Monitoring on a New Microsatellite. It's much easier to remember Venus. So it's an Earth observing satellite. And it's a joint French-Israeli collaboration, tens of millions of euro or dollars involved. And it was launched in August 2017, 3.5-year mission. Its orbit started off at about 720 kilometers up, and it dropped down to 410 kilometers. That means we have higher resolution data. It has what's called a near polar sun-synchronous orbit. It just follows the sun around. It collects information on from 12 spectral bands, and these are just 12 different wavelengths. It's kind of multispectral. Now that's really useful because these different spectral bands give different information about a wide range of environmental factors, and some of them are vegetation. It has a 2-day revisit time, and it has a 5 to 10 meter resolution. You saw the picture earlier with the little pixels. We have information for every one of those little pixels, and we've got 120 special sites of interest. So these are the sites of interest. There's Milwaukee, Downer Woods right in the middle of them. You can see there's quite widespread on all continents and all locations. Now, we're the only ones looking at phenology. The other ones would be looking at things like clouds and aerosols and air pollution or water pollution or soil or like a load of things that we wouldn't even understand. But anyway, so we're the phenology people working on this, and this is just a very far image of Downer Woods there. Okay, so to compare Venus information with standard satellite data that we have. So this is the image of Downer Woods. This is a MODIS pixel, and these are all the tiny little Venus pixels. So what we have here is the satellite, our Venus satellite. The spatial resolution is 5 to 10 meters, and for MODIS, it's 250 meters. The smallest resolution is 250 meters. So it means that we have information for Downer Woods at this scale, but we only have one number for our vegetation index at this scale. Whereas you can see there's a lot more detail available in the higher spatial resolution of the Venus images. So, that was important to us. Venus goes by every 2 days, which is great for phenology. Because some of the other satellites like Landsat go by every 16 days. And if there's cloud cover, that means you would only get information once a month. So that's not great for phenology work. The global coverage is about 120 sites and only from 2017, whereas the MODIS, it has global coverage and we have information from the year 2000. Now these other satellites, well, they also provide information as well, but just slightly different they have just different types of sensors. The information we get are vegetation indices. Now, vegetation index is just giving us information about the reflectance of the absorption of red light and the reflectance of infrared light. So it just means that it gives us information on whether the vegetation is healthy or not, or whether it's green or not. So we're looking for greenness. I know we're measuring near infrared and red, but it tells us whether the leaves are healthy or not. Okay, vegetation does reflect some green, but it mostly reflects in the near infrared and it absorbs the red. So what's happening is that the index then is a measure of the relative importance of those two characteristics. there are lots of other different types of indices. Like all of these different indices, they just use a slightly different way of calculating what we're seeing on the ground, the different vegetation indices. And they evolve over time, you know, when they get better technology and better methods to detect what's going on the ground. But, well, anyway, we just have a wide range of different indices. And this is what we have. This is the vegetation indices, the 5 different vegetation indices that are available from the MODIS, or from the Venus satellite, at this very high spatial resolution. Now, here we have Downer Woods, and you can see this is the 10-meter pixels. So each of these pixels is giving us some information. And what we're seeing is the data here. I'll just show the EVI. This is just called Enhanced Vegetation Index, Near Infrared Vegetation, Normalized Different Vegetation. They're just different types of indices, but they use the same red and near infrared bands. Some of them use other spectral bands as well, but let's just stick with these 2. So what we expect to see is, we expect to see that the vegetation index is low during the winter months, and then it increases as the leaves come out and we have a higher rate of photosynthesis and we can get more greening, and then it kind of levels off. There's lots of issues with the data, but it kind of levels off and then heads down as the greenness goes down in autumn. And we have that for the 2 years. Like, you have no idea the amount of processing of this data that goes on behind the scenes. We don't have the data for 2020 yet available to us, but the people who run the Venus project, they process the data, they make it available to us in a raw data format, and then we have to pre-process it, and we have lots of issues with the data. There are lots and lots of issues with missing data and missing images, and lots of cloud cover means that we can't really see what's going on in Downer Woods, and that causes huge problems. But overall, we didn't really find any significant relationship with the phenology that we observed in Downer Woods on the ground. So, but this is the kind of images that we get, and we can see this is the average of all the little boxes over the site, and it goes up and down in spring and then down in autumn. And all the vegetation indices show a similar, they track the similar trend. So for Downer Woods, we have a wide range of different methods that we observe the data for. And the difference in what we're seeing on the ground and what we see from the camera is based really on the view angle. When we're in Downer Woods, we're looking up into the canopy for the trees, for example. When the canopy is closed, the satellites or the PHENOCAM cannot see the shrubs. So they're looking down. So we can see here, this is a PHENOCAM image looking down on the woodland. So with direct observations, we're only looking at individual trees and shrubs. We monitor these different phenophases. The PHENOCAM takes an image every 30 minutes, and then we analyze the greenness of that. The PHENOCAM also gives us information on what's called the green chromatic coordinates. And then we can get a time series. So this is what we would have expected to see with the Venus data, like a lot of data going up in spring and then coming down during the summer and then really decreasing in autumn. Now, the reason I've just marked this 23rd of October is because according to the PHENOCAM, the green chromatic coordinate saying very, very, very little greening. We're observing the shrubs and we're saying a lot of greening. The PHENOCAM image is showing some greening, and then the satellite data is showing a bit of greening as well. So the point is that we have all these different methods of observing the same thing, and we're not seeing, you know, we're all observing slightly different things. Like, we're absolutely observing what's going on at the ground. The image is taking the top of the canopy, and then the green chromatic coordinate is analyzing the time series or the phenological profile over time. And then again, of course, the satellite is looking on the top. So matching what's going on with remote sensing and in situ phenometrics is quite tricky. Oh yeah, I wanted to talk a little bit about chlorophyll. So I was saying that the chlorophyll in the leaves, so we know that the shrubs are staying greener for longer, but are they photosynthesizing at the same time? So what we do, this is Chloe, she goes out and uses this little meter, and the meter just gives you an indication of how much chlorophyll is in the leaf. It's a non-destructive, handheld little device, and it's very, very useful. The higher the number, 47, very high amount of chlorophyll. If it goes down to about 10, which means there's not very little chlorophyll. This is Erica Young, and she's measuring the activity of the chlorophyll. So whether we have chlorophyll, yes or no, and then how active it is. And we got some very, very interesting results that I'm just going to show you now. So this is a temperature trend in Downlow Woods during the autumn of 2018. And I mean, yes, lots of variation in the temperature, but there's a sudden change in the regime here at day of the year 285, about the 11th of October. We see a clear shift to cooler temperatures from 16 degrees to 6 degrees Celsius. So if we look, this is just the rate of photosynthesis. So we have native species and non-native species, and they're photosynthesizing pretty high. And there's not a huge difference between the native and the non-native. Non-native tend to be a little bit higher rate of photosynthesis. But look what happens at this point. We see a very dramatic decrease in the photosynthetic rate of both the native and the non-native, sorry, the native with the dark color and the non-native, and also greater variation. So it means the chlorophyll was, the photosynthetic rate within the chlorophyll was like changed a lot. It decreased, and there was a lot of variation within that decrease. But if you look at the total chlorophyll within the leaf, we also determine the total chlorophyll within the leaf. You can see there's the general decline over the season, which is what you'd expect in autumn. For the natives and the non-natives, there's less chlorophyll in the non-natives in general than in the natives than in the non-natives. So the invasive species have more chlorophyll, but there's no detectable shift in the pigment. There's a shift in the activity of the pigment, but not in the pigment itself. And the same with the SPAD meter. This is just a little chlorophyll device. So what we're seeing is that the activity of the chlorophyll is really impacted by a change in temperature, but the actual chlorophyll itself is not changing. So there's a change in the photosynthetic rate. So we see this in autumn. That's very important for the timing of the active photosynthetic period when carbon dioxide is being extracted from the environment. So just to finish off, I wanted to talk a little bit about the discrepancies we see between the in situ data, what we're monitoring on the ground, and the satellite remote sensing, and why we see these differences. So for example, with in situ data, we only have a small number of trees that we can monitor, whereas the remote sensing can look at vast areas, a landscape area or larger. We have a fairly long-term series for the in situ data, but a very short-term series for the remote sensing. The view angle is important. This is a picture I took down near the lake during the autumn. And if we're monitoring from the ground, we're observing total greenness. This tree, we would say, would be totally green. Whereas from above, the satellite or the remote sensing would be saying it's totally yellow or totally colored. In situ, we're limited to spring and autumn, whereas with the remote sensing, we have continuous year round. We can be very subjective in our observations, whereas the satellites are usually not. They're very accurate about how much green there is or how much red there is. But with the remote sensing, we have a range of different types of sensors. And then we have different definitions of phenophases, and the validation of remote sensing can also be quite complex. We need the in situ observations to validate what we're seeing with the satellites. So just to quickly sum up, we've seen that the shrubs have an extended growing season compared to the trees. We see there's a difference in the non-native and the native vegetation, and it's particularly in the phenological profiling of the two groups. The Venus data is promising, we haven't gotten there yet, but it is definitely promising. We still have a lot of work to do, and obviously the project is not finished. There's lots of gaps and challenges, and we need to compare the Venus data with the MODIS and the PHENOCAN. We have a lot more work to do on that. And I just wanted to thank the Research Growth Initiative grant from the University of Wisconsin-Milwaukee, which provided the funding for this project, and also, of course, to Dennis for protecting the woodlands for us. So I'm going to end there and thank you all for your attention. And if there are any questions, I think we can take them.
Our first question is, have you noticed any difference in phenology for the shrubs and trees in the shadow from the Sandberg towers?
We can observe, no, we don't really, because the shadow moves. It's not fixed. So we would have the shadow in the Sandberg towers just for a couple of hours during the day. And that's not enough to significantly impact the phenology in those sites. But it's a very good question, actually, yeah, because we do have influences from the, like if the woodland itself being within a city, it's slightly warmer than the natural environment further out. But no, they don't really have an impact. We can see small changes in the temperature, but it's not enough to impact the phenology. Great question.
And how do you choose the plants you observe? Do you have capacity to observe more in the winter months? From Wyatt.
We choose the plants based on the abundance and the convenience of the plants. We'd like to be able to do more. We go out in the field and we decide, or into the woodlands, and we decide what species we can observe, and then we select individuals, up to 5 individuals of each particular plant, and then we observe that every other day. And then, of course, we have issues like I've shown you earlier with damage to the plants, and we have to select new ones. But we only have 2 seasons when we can observe. Like, we just don't have the time to go out every single day and take observations, but we do go out every other day during campaigns in the spring and in the autumn at the extremes of the seasons. So there's not much happening really in winter. Okay.
Okay, since chlorophyll activity decreases with temperature, even while leaves still appear green, does that mean visual observation, whether sensors or humans, would overestimate carbon sequestration in autumn? From very astute question from Trevor.
Yes, Trevor, exactly. We would. That's why we wanted to try and quantify that, to see how the activity. Yes, just because they're green doesn't mean that they're photosynthesizing. However, we can also get some information on photosynthetic activity from satellite data, just not from the satellite data that we have. Like, there's another part of the spectrum called the photosynthetically active radiation. So that's radiation that the plants are using. And we can measure that photosynthetically active radiation using satellite data. And we can get an idea of how active different ecosystems are. So it helps in that way, yeah, but definitely it can overestimate the carbon sequestration in autumn, except there is, as the technology develops, we get more and more information. So now we have to use the greening and this FA, the fraction of absorbed photosynthetically active radiation also. But yeah, nice question.
Are you using or encouraging farmers to use organic practices to maintain and protect the vegetation? From Alan.
It's a little bit outside of my wheelhouse, really. We do encourage people to practice organic or to practice organic farming. And even in the city, we see people doing organic gardening and using less water also to try and maintain our water supply and to try and keep the environment as clean as possible and to try and reduce the amount of herbicides and pesticides and fertilizers that we use. So we would certainly be advocates of protecting the environment in any way we can. And because agriculture is so, or has the potential to, well, provide lots of food, but also has the potential to pollute. I think now that people are more aware of timing their practices, such as applying herbicides and fertilizer at different times and insecticides also. Like if you get the timing right, you can reduce the amount of damage that can occur. Yeah, great question.
Okay, we have a question from Joe who asks, out of curiosity, why isn't the plot of land to the east at the corner of Downer and Edgewood included as part of the Downer Woods?
Why isn't the plot of land at the east? I have no idea. It is still part of Downer Woods, but it's not the part that's fenced off. So the part that's fenced off is the bit that we use for our research and for our teaching. But I have absolutely no idea why it's not included within the fence. Sorry.
You can't know everything. Okay, let's see if we have any additional questions here. Ellison, I'm curious while we wait to see if there's an additional question. How do you see that the Downer Woods is well utilized for recreation by students and faculty and staff since you're out there so much?
Yes, we see a lot of activity in Downer Woods. A lot of people walk through the woods during their breaks. I've seen students up in the trees. I met one student once up in the trees playing a tin whistle. There was a gentleman that I used to meet, and he would really put the logs around the footpath so people wouldn't, walk off into the woods. He really maintained the structure of the paths as well. And then I see a lot of people, walking their dogs, which is not really allowed, but and yeah, it's used a lot. And I see some kids also make forts and things in the woods. So it's extremely well used, really well used.
That's a great resource in that little urban pocket of campus to have that.
Yeah, and we use it a lot for teaching purposes as well. I mean, it's one thing to tell students the 3 different levels of vegetation in a forest. It's quite abstract, but when you can bring them out and they see it, and they see the different types of trees and the different shrubs and how they interact, it's very, very useful.
Okay, so this is our final question. Do you have an ideal outcome for the overlap of your observations and the tools from space?
We would like to be able to determine exactly what phenophases the satellite is observing. Ideally, and then we could extrapolate that to areas like far from Downer Woods. But the more information we collect and the more we look at the satellite data as well, and because basically we're observing different things, the satellites are observing green in principle and photosynthesis now, and we're observing on the ground like individual trees. And it's very hard to reconcile those 2 factors. But if we can get enough information and enough data and a longer time series, we will be able to use it for general purposes, like in planning, definitely, and to try and figure out the carbon budgets. I mean, it's better than what we have at the moment. So ideally, we'd like to be able to say exactly what's happening, and it will happen. The satellite, the resolution of the satellites is improving. What they're seeing is improving, and what we're monitoring on the ground also will help that as well. So that would be nice.
Great. Thank you so much again for sharing your research with us and joining us. It's fascinating to hear everything that's happening inside the woods that we all pass on a daily basis. So thank you for everybody for joining us today. We had a really nice attendance, and we look forward to hopefully seeing you at the next Master Chat coming up February Thursday 18th. We're featuring Professor Clark Evans, who will share a presentation called Hurricanes 101, the why and the how applied to the hurricane season of 2020. So hopefully I'll see you then. Thank you.
Thank you, Amy.